Hello everyone, and welcome. I'm excited to have you here today. Today, we're going to explore photosynthesis in higher plants. This is the title of our presentation, and it promises to be both informative and interesting. Over the next few slides, we'll cover how plants convert sunlight into energy, the process itself, and its importance to life on Earth. Let's get started. Let's begin our exploration of photosynthesis with an overview of its key aspects. On this slide, we see that the process is broken down into several important components. First, we have the splitting of water, which is a crucial step in providing electrons and protons for the process. Then, we look at the pigments involved in photosynthesis, mainly chlorophyll, which absorb light energy effectively. Moving on, we consider how ATP and NADPH are used. They serve as energy carriers that power the synthesis of sugars. Next, we examine the electron transport system, a series of proteins that transfer electrons and help generate the energy-rich molecules. The site of photosynthesis is primarily in the chloroplasts of plant cells, specifically in structures called thylakoid membranes. Factors affecting photosynthesis, such as light intensity, carbon dioxide levels, and temperature, can influence the efficiency of the process. We also explore the chemiosmotic hypothesis, which explains how ATP is produced through a flow of protons across membranes. The C4 pathway is a specialized adaptation that allows certain plants to photosynthesize efficiently under high light and temperature conditions. The overall process of photosynthesis involves capturing light energy, converting it into chemical energy, and synthesizing glucose. Photophosphorylation refers to the formation of ATP during the light-dependent reactions. To better understand how this all began, we look at early experiments that laid the foundation for our current knowledge. Finally, we consider what exactly happens during the light reactions, which are the first step in this complex but beautifully orchestrated process. All these points connect to give us a comprehensive picture of how photosynthesis sustains life on Earth. Photosynthesis produces oxygen as a byproduct, which is crucial for most living organisms on Earth. When plants and other photosynthetic organisms carry out this process, they release O2 into the atmosphere, helping to sustain life. But beyond oxygen production, photosynthesis also serves as the fundamental source of energy for nearly all living things. It's essentially the foundation of the food chain. At its core, photosynthesis is an enzyme-regulated anabolic process. It involves the manufacture of organic compounds, mainly sugars, inside the chlorophyll-containing cells. This process uses carbon dioxide and water, with sunlight acting as the energy source. It's a fascinating chemical process where light energy is transformed into the chemical energy that fuels life. So, in summary, photosynthesis isn't just about making food for plants. It's the key process that supports almost all life on Earth by providing energy and oxygen. Let's discuss some of the early experiments that helped us understand photosynthesis. Cornelius van Niel, for instance, performed experiments with purple and green bacteria. He demonstrated that photosynthesis depends on light, and he explained that hydrogen from water reduces carbon dioxide to form carbohydrates. An important conclusion he made was that the oxygen produced actually comes from water, not from carbon dioxide itself. This led to the formulation of the correct overall equation for photosynthesis, where we see that carbohydrates, represented as CH2O, are produced along with oxygen and water. Moving to the 19th century, scientists first explained key features of photosynthesis. They understood that plants use light energy to convert carbon dioxide and water into carbohydrates. The empirical equation we now associate with this process sums it up. Carbon dioxide plus water, in the presence of light, produces glucose and oxygen. In 1854, Julius von Sachs made a significant breakthrough by showing that plants produce glucose as they grow, storing it as starch. He found that plants contain a green substance, chlorophyll, located within specialized cell bodies, mainly in the green parts of plants. These green parts are where glucose is synthesized. T.W. Engelmann's experiments with a prism split light into the spectrum of colors, what we call VIBGYOR. He observed that green algae like cladophora, when placed in a suspension of bacteria, attracted bacteria mainly in areas exposed to blue and red light. This helped explain how chlorophyll absorbs certain wavelengths, specifically chlorophyll A and B. Jan Genhouse, another key figure, showed that sunlight is essential for plants to purify the air. He conducted experiments with aquatic plants, and in bright sunlight, small bubbles, oxygen, formed around the green parts. In darkness, they didn't. So he proved that only the green parts of plants are responsible for releasing oxygen. Finally, Joseph Priestley's experiments with a bell jar, a candle, and a mouse revealed that burning candles or animals deplete the air. But when he introduced a mint plant, it kept the air fresh enough for the candle to burn and the mouse to survive. This led him to propose that plants restore the air by releasing something, what we now know as oxygen, that is consumed by animals and combustion. All these experiments laid the groundwork for our modern understanding of photosynthesis, showing how plants capture light energy and convert it into chemical energy, producing oxygen and carbohydrates in the process. Photosynthesis mainly happens in the green parts of the plant, and most of the process takes place in the leaves. Inside a leaf, the key players are the mesophyll cells, which contain tiny structures called chloroplasts. These chloroplasts are actually where the magic of photosynthesis happens. Within the chloroplasts, there are thylakoids. These are membrane-bound structures that contain the pigments we need to capture sunlight. These pigments, like chlorophyll, are essential because they absorb solar energy to kick off the whole process. The thylakoids are organized into stacks called grana, and these are crucial because they trap light energy and help produce the energy molecules ATP and NADPH, which power the later stages of photosynthesis. Now the next phase, the biosynthetic or dark reaction, takes place in the chloroplast stroma, which is like the fluid-filled space inside the chloroplast. Here, carbon dioxide is reduced to form carbohydrates, which are the sugars the plant uses for energy and growth. So, in summary, the entire process is localized specifically in the green parts, mainly leaves, with specialized structures inside the chloroplasts working together to convert sunlight into usable chemical energy. This localization is a fundamental aspect of how plants efficiently carry out photosynthesis and support the entire food chain. Let's take a closer look at the pigments involved in photosynthesis. First, we see the range from yellow to yellow-orange, which corresponds to carotenoids. These pigments play a crucial role in capturing light energy. Then, there's yellow, which is characteristic of xanthophylls, another type of carotenoid. Moving on, you'll notice that in the blue and red regions of the spectrum, photosynthesis occurs at higher rates. This is because chlorophyll absorbs light most efficiently in these regions, making them essential for energy capture. Now, looking at yellow-green, we see chlorophyll. In chromatographs, chlorophyll B appears bright or blue-green, and it's a major pigment in photosynthesis. It acts as a reaction center, helping trap and convert light into chemical energy. Finally, we have chlorophyll A, which is the primary pigment that drives the process. It absorbs light directly and initiates the energy transfer necessary for photosynthesis to occur. Together, these pigments work harmoniously to maximize light absorption across different wavelengths, ensuring the plant can efficiently harness solar energy. On this slide, we're looking at the key components involved in the light reactions of photosynthesis. First, you'll notice that chlorophyll A has specific absorption peaks, one at 680 nanometers, known as P680, which is associated with photosystem II, and another at 700 nanometers, called P700. Linked to photosystem I, these pigments are essential because they absorb different wavelengths of light, making the process more efficient. Each photosystem contains pigments bound to proteins, except for one special chlorophyll, a molecule that forms what we call the reaction center. This reaction center is where the magic happens, where the absorbed light energy gets converted into chemical energy. Surrounding these reaction centers are light harvesting complexes, or LHCs, made up of hundreds of pigment molecules attached to proteins. These complexes are designed to capture light across a broad spectrum and funnel that energy efficiently to the reaction centers. Now, this entire process takes place only in the presence of light and is often called the light reactions or the photochemical phase. During this phase, we see several critical steps: light absorption, the splitting of water molecules, which releases oxygen, and the formation of high-energy compounds like ATP and NADPH that power the next stages of photosynthesis. In essence, these light reactions are fundamental because they harness light energy to produce the energy carriers that fuel the entire process of converting sunlight into chemical energy. Understanding this helps us appreciate how plants efficiently capture sunlight and turn it into the energy needed for growth and survival. Now let's talk about the light-independent phase, also known as the biosynthetic phase or the dark reaction. This part of photosynthesis is all about synthesizing food material, specifically sugars, without the direct need for light. It's like the plant's way of assembling its food after capturing sunlight. During this phase, the plant uses the energy stored in ATP and NADPH. These are energy carriers produced earlier in the light reactions to convert carbon dioxide into glucose and other sugars. This process is crucial because it allows the plant to store energy in a form that can be used later for growth and development. It's important to note that while this phase doesn't require light directly, it depends heavily on the products generated during the light-dependent reactions. This interconnectedness highlights how both phases work together seamlessly to sustain plant life. Understanding this biosynthetic phase helps us appreciate how plants convert simple molecules into complex carbohydrates, supporting not just their own growth, but also providing the foundation for most food chains on Earth. During the transport of electrons in photosynthesis, what actually happens is that electrons are transferred starting from photosystem II, or PSII. The electrons move uphill to an acceptor, then travel down the electron transport chain to photosystem I, or PSI along this path. Electrons get excited, transferred to other acceptors, and finally move downhill to reduce NADP+ into NADPH plus H+. This entire process is called the Z scheme because of the shape it forms, which looks like a Z when you place all the carriers in sequence on a redox potential scale. Now, an important point to remember is that electrons are not consumed as they pass through the chain. Instead, they are passed on to the pigments in PSI. When electrons arrive at the reaction center of PSI, they're excited again by red light at 700 nanometers wavelength. These excited electrons are transferred to another acceptor molecule with a higher redox potential. The hole left in PSI is then filled by electrons coming from PSII, maintaining the flow. From PSI, electrons continue downhill, reducing NADP+ to NADPH plus H+. Focusing on the photosystem center, chlorophyll absorbs light at 680 nanometers, which excites electrons and causes two to be released from the atomic nucleus. These electrons are captured by a primary electron acceptor called ferredoxin. Then, the electrons move through the electron transport system, which involves cytochromes, in a series of oxidation-reduction reactions. As they move downhill in redox potential, they transfer energy efficiently until they finally reach PSI again. This entire system is crucial because it provides the reducing power needed for the Calvin cycle and ultimately drives the synthesis of sugars in photosynthesis. Let's take a closer look at how the electrons needed for the electron transport system are actually supplied. This process is called photolysis, and it occurs specifically at photosystem II. Essentially, PSII constantly loses electrons, which are then replenished by the electrons released during the splitting of water molecules. This splitting of water, or photolysis, happens in the presence of light, which provides the energy needed for the process. It involves key elements like manganese ions, Mn2+, and chloride ions, Cl, which help facilitate the reaction. When water is split, it produces three important products: hydrogen ions, or H+, oxygen gas, O2, and electrons. The oxygen that's released is actually a byproduct of photosynthesis, which is pretty fascinating. It's the oxygen we breathe and rely on. And, importantly, this entire process takes place right near photosystem II, ensuring a continuous supply of electrons to keep the whole photosynthetic system running smoothly. Let's begin by understanding cyclic photophosphorylation. In this process, only photosystem I, or PSI, is involved. The electrons excited in PSI circulate within the same photosystem, creating a sort of loop. This process primarily takes place in the stroma lamellae, which is a specific part of the chloroplast. The reason for this is that in this region, PSII and the NADP reductase enzyme are absent, so only ATP is produced here. Now, moving on to non-cyclic photophosphorylation, this involves both photosystem II and photosystem I working in series. The electrons flow from PSII to Psi through an electron transport chain, which is often called the Z-scheme because of its shape. This process is essential because it results in the synthesis of both ATP and NADPH plus H+. Both PSII and Psi are located in the lamellae of the grana, which are the stacked regions of the chloroplast. That's why this entire process occurs in those regions. To connect everything, photophosphorylation refers to the process of synthesizing ATP from ADP and inorganic phosphate, and this happens in the presence of light. It's a way for living organisms to extract energy from light-dependent reactions, storing it in high-energy molecules like ATP and NADPH. And remember, this process occurs inside cells, specifically in mitochondria and chloroplasts, which are the energy hubs of the cell. There are two main types of photophosphorylation: cyclic, which we've just discussed, and non-cyclic, which involves both photosystems. Both are crucial for the overall process of photosynthesis, providing the energy and reducing power the plant needs to synthesize sugars and other vital compounds. NADP reductase enzyme is located on the stroma side of the membrane, along with electrons from the acceptor. It removes H+ from the stroma during the reduction of NADPH plus H+. This process creates proton gradients across the thylakoid membrane, which is really important for energy transfer. It also causes a measurable decrease in pH in the lumen, meaning the inside of the thylakoid becomes more acidic. Elcaas has a channel that allows protons to diffuse back to the stroma across the membrane. As these protons flow through, energy is released, which activates the Elcaas enzyme. This enzyme then catalyzes the formation of ATP, which is essential for the plant's energy needs. Chemiosmosis, as a process, requires a membrane, a proton pump, a proton gradient, and Elcaas. The energy from sunlight helps pump protons across the membrane, creating a high concentration of protons inside the thylakoid lumen. This gradient is the driving force for ATP production. The chemiosmotic hypothesis explains how ATP synthesis is linked to this proton gradient across the thylakoid membrane. Essentially, protons accumulate inside the lumen as a result of water splitting and electron transport. As electrons move through the photosystem, they help transport protons across the membrane, reinforcing the gradient. This proton buildup inside the lumen provides the potential energy needed for ATP synthesis. So, the movement of protons back through Elcaas releases energy, which is then used to produce ATP. This whole process demonstrates how the energy from sunlight is converted into chemical energy, powering the plant's metabolic activities. Now, let's talk about how ATP and NADPH are used in the process of photosynthesis. These molecules are essentially the energy carriers produced during the light reactions. Once they are formed, they don't just stay in the chloroplast. They're actively involved in the next stage, which is the biosynthetic phase. This is where the plant actually synthesizes sugars and other organic molecules. ATP provides the energy needed to drive various enzymatic reactions. Think of it as the fuel that powers the process. NADPH, on the other hand, supplies the reducing power, meaning it donates electrons necessary for converting carbon dioxide into organic molecules. It's important to note that these reactions happen in the dark as well, because they depend on ATP and NADPH generated during the light-dependent reactions, not directly on light itself. So, the presence of light is crucial indirectly, because it produces these energy-rich molecules. And of course, CO2 and H2O are the raw materials, but without ATP and NADPH, the plant wouldn't be able to turn those raw materials into sugars. This interconnected system highlights how the light reactions and the biosynthetic phase work hand in hand to ultimately produce the food that sustains the plant and us. Let's now talk about the regeneration of the CO2 acceptor molecule, which is crucial for the cycle to keep going smoothly. Essentially, after the cycle uses up Rhu-BP, it needs to be regenerated so that the process can continue without interruption. This regeneration step requires energy in the form of ATP, specifically one ATP molecule for phosphorylation to recreate Rhu-BP. It's important to remember that for every CO2 molecule that enters the Calvin cycle, we need 3 molecules of ATP and 2 molecules of NADPH. This highlights how energy-intensive the process is. Now, when we consider how a glucose molecule is formed, it takes 6 CO2 molecules and 6 turns of the cycle. These 6 cycles lead to the formation of glucose, involving a series of reactions that utilize 2 ATP molecules for phosphorylation and 2 NADPH molecules for reduction per CO2 fixed. Let's also touch on the reduction phase, where CO2 is fixed into a stable organic compound. This happens through a process called carboxylation, where CO2 is attached to Rhu-BP, forming 2 molecules of 3-PGA. This reaction is catalyzed by the enzyme Rhu-BP carboxylase, commonly known as Rhu-bis-Co, which interestingly has a dual role. It can also catalyze oxygenation reactions. And finally, the entire Calvin cycle operates in a cyclic manner, as proposed by Melvin Calvin and his colleagues. This cycle not only fixes CO2, but also regenerates Rhu-BP, allowing the process to continue seamlessly. It's a universal pathway in all photosynthetic plants, regardless of whether they follow the C3, C4, or other pathways. So, in essence, the Calvin cycle is a beautifully integrated process that converts inorganic CO2 into organic molecules, driven by energy from ATP and NADPH, and sustained by the regeneration of Rhu-BP. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is then transported back to the mesophyll cells, where it is converted back into PEP, completing the cycle. The CO2 released in the bundle sheath cells enters the C3, or Calvin pathway, which is a process common to all plants. Interestingly, the bundle sheath cells are rich in the enzyme ribulose bisphosphate carboxylase oxygenase, known as Rhu-bis-Co, but they lack PEPCase. This means that the basic pathway leading to sugar formation, the Calvin cycle, is shared by both C3 and C4 plants. Now, the tissues involved in the C4 pathway are primarily the mesophyll and bundle sheath cells. The process begins in the mesophyll cells, where CO2 is accepted by a primary acceptor molecule called phosphonol pyruvate, or PEP for short. This reaction is catalyzed by PEP carboxylase, or PEPCase. It's important to note that mesophyll cells don't contain Rhu-bis-Co. Instead, they form a C4 acid, oxaloacetic acid, or OAA, which then produces other 4-carbon compounds like malic acid or aspartic acid. These compounds, especially malic acid, are transported to the bundle sheath cells. The pathway that C4 plants follow is known as the Hatch and Slack pathway, and it's a cyclic process, meaning it keeps regenerating itself. Now, anatomically, C4 plants differ quite a bit from C3 plants. They have large cells called bundle sheath cells surrounding their vascular bundles, a feature known as Krantz anatomy. These leaves often have several layers of these cells, which are packed with chloroplasts, and they have thick walls that are almost impervious to gases. Interestingly, they lack intercellular spaces, which helps in maintaining a controlled environment for the pathway. C4 plants are specially adapted to hot, dry environments. They tolerate higher temperatures, respond to more intense sunlight, and they don't undergo photorespiration, which is a wasteful process in C3 plants. Because of these adaptations, they tend to produce more biomass, making them highly productive. Finally, plants that thrive in dry tropical regions often use the C4 pathway. The initial CoA2 fixation product here is C4-oxaloacetic acid. Despite these differences, C4 plants still follow the Calvin cycle as their main pathway for biosynthesis, integrating this efficient pathway to maximize productivity under challenging conditions. Water's effect on plants is more indirect than you might think. When plants experience water stress, they respond by closing their stomata, which are tiny pores on the leaf surface. This reduces the amount of CoA2 that enters the leaf, ultimately limiting photosynthesis. Additionally, water stress causes leaves to wilt, decreasing their surface area and slowing down metabolic activity within the leaves. So, water availability influences photosynthesis mainly through these physiological changes, rather than a direct chemical impact. Now, moving on to carbon dioxide, which is a major limiting factor for photosynthesis. It's important to remember that CoA2 is present in the atmosphere at very low concentrations, roughly 0.03 to 0.04%. Interestingly, increasing CoA2 levels up to about 0.05% can boost the rate of carbon fixation, helping plants photosynthesize more efficiently. However, if CoA2 levels go beyond this point for prolonged periods, it can actually become harmful, potentially damaging the plant's processes. Next, let's talk about temperature. The dark reactions in photosynthesis are enzyme-driven, so they are quite sensitive to temperature changes. Light reactions are also affected, but to a lesser extent. C4 plants, for example, typically have a higher temperature optimum compared to C3 plants. The ideal temperature for photosynthesis varies depending on the plant's natural habitat. Tropical plants, which are adapted to warmer environments, generally have a higher temperature optimum than temperate plants. When it comes to light intensity, at low levels, the relationship between incident light and carbon fixation is pretty much linear. More light means more photosynthesis. But at higher light intensities, the rate levels off because other factors become limiting. If light intensity increases beyond a certain point, it can actually harm the plant by breaking down chlorophyll, leading to a decrease in photosynthesis efficiency. And finally, I want to highlight Blackman's law of limiting factors. It states that if a process is affected by multiple factors, the overall rate is determined by the factor closest to its minimal level. In other words, the process is only as fast as its most limiting factor. External factors like sunlight, temperature, CoA2 concentration, and water availability all play crucial roles in regulating photosynthesis. Understanding these interactions helps us see how plants adapt and respond to their environment, guiding us in optimizing conditions for better plant growth and productivity.